All right. Thank you, everyone. I hope you enjoyed your coffee break. We're going to go ahead and get started. So it's my great pleasure today to introduce Dr. Paul Mead. He is the principal scientist in the Department of Pathology at St. Jude Children's Research Hospital. I can say that I've seen Dr. Mead speak before. This is going to be absolutely wonderful, and we're looking forward to hearing him talk about full spectral flow and his lab. Welcome, Dr. Mead.
Oh, thank you very much for that very kind introduction, and thank you for the opportunity to come and speak here today. All right, let's see if I can get through all of these slides. Okay, so I'm from St. Jude Children's Research Hospital, which is in Memphis, Tennessee. For those of you who don't know, St. Jude is a non-profit hospital, and we're dedicated solely to the treatment of kids with catastrophic diseases. We were the first NIH-designated comprehensive cancer center that is solely dedicated to treating pediatrics. We have just over 5,000 employees, and as our sort of name implies, we're half clinical and half research. And one of the really nice things about St. Jude is that families never get a bill from the hospital. Okay, so working for a non-profit, you tend to accumulate job descriptions. So one of my jobs is that I am director of the translational immunopathology lab, and our lab is tasked really mainly with developing new tests for the clinical lab and also doing clinical trial flow, so data that will not end up in the patient record. We do that in this lab over here. Now, my other role is that I'm also nominally the technical director of the clinical immunopathology lab. So I'm also running the sort of clinical side as well. And I sort of liaise between, and it kind of makes sense because we develop tests in one lab and we do the analysis and clinical testing over in the clinical lab. I took this view of St. Jude because actually you can see my house from there. Anyway. So I don't get out much. That's sort of the bounds of my roaming, as it were. I have no conflicts of interest to declare. And pretty much for the first half of the talk, I'm going to talk about the work of one really talented physician scientist in my lab, Pei-Ting Liu. He's done pretty much all of the work that you'll see on the development of these high parameter spectral panels that we're going to be seeing. So there's Pei-Ting. So at St. Jude, and pretty common for any sort of leukemia service, we get a patient that comes in and we take a sample. And this is sort of the overview of the at diagnosis, these sort of standard sort of labs that would see samples and a type of testing that we would do. And today, of course, what we'll be interested in is the immunopathology, the flow lab. And we generally get the second tap, but the second drawer of a bone marrow would come to the flow lab for testing and immunophenotyping. So our standard workflow for a new or relapsed leukemia would be that we'd have this patient sample would come in. We would first do immunophenotyping. After that, we would determine the lineage of that leukemia. And then we would also, that's on day one or day zero, whatever you would call that diagnosis, we would then run a B, T, or AML MRD panel to be able to determine the baseline of MRD and also determine what markers we need to use to follow that leukemia during follow-up treatment stages. So at St. Jude, our upfront immunophenotyping panel is quite extensive. This is done on the Canto 2, a 3-laser 8-color instrument. Our immunophenotyping panel consists of 13 different tubes. We have over 44 surface markers and 4 intracellular markers, so it's really quite extensive, probably more than you actually need just to determine whether it's a B, T, or an AML. But there's a lot of more information in there. And again, this has all been run for the last 12 years or more on the BD Canto 2 3-laser 8-color cytometers. So upon lineage assignment, say for example, this little blue chap up here has a BALL, we would then at diagnosis on that same sample also run the B MRD panel. And we'll go through this in a little bit of detail because it will come up later on and sort of give you an idea why we wanted to move to a high parameter flow platform such as spectral. So all of our MRD panels are based around these backbones of markers, and these really allow us to identify our blast population. So for the B, our backbone essentially is CD45, 19, 22, CD10, and 34. And then we have these add-on markers which we use to identify a leukemia-associated immunophenotype that we will use for that specific patient to be able to follow the leukemia and during follow-up treatment time courses at later stages. And we'll see this in a few minutes. So here's just an example of what that would look like. Here is our backbone markers, and you can see we have a population of CD45 dim, 19 positive blasts. They uniformly overexpress CD10 and also positive for CD34. So clearly a blast population. So then we use our add-on markers, those 3 other channels on the Canto 2, to be able to determine what is the immunophenotype of this patient that we can use to follow later on. And this one would be relatively simple. We see it's got dim 38, overexpression of CD73, and dim CD45. So all of these other gates that you can see here are sort of normal gates that we've used, where we've taken normal regenerating marrows from a dozen or more different patients and drawn the sort of 95% confidence limits of where normal regenerating B cells would be, or hematogones. And so we can see, and these are really just a visual guide for our hematopathologists to be able to determine, you know, which ones are well outside those. So they may be candidates for you doing for our immunophenotype that we're going to follow for that patient. So for this kid, the best markers to use would be CD38, 73, CD20, and again on that backbone. Oop. There we go. So by the end of the diagnostic day, we would have run quite a large number of tubes, right? So we've run at least 13 tubes here, and another 9 tubes for the MRD diagnosis, all to end up with a patient-specific MRD follow-up panel that we run for this individual patient at every treatment time point thereafter. So you can imagine it's a lot of work doing this, a lot of tubes, but also whenever we get a sample from this patient, the techs in the lab also need to know that this is the panel that gets run with this patient. So there's a lot of pre-analytic work to make sure that the right patient gets the right panel, etc. So you can imagine there's a lot of work involved. And at St. Jude, we see around 300 new or relapsed leukemias a year, and we see about 1,600 to 2,000 of these follow-ups. So it's quite a busy service in our pediatric leukemia population. Now, as I've mentioned, we have been running this for the last 12 years or so on the BD 3 laser 8 color Canto 2. Now, these are reaching the end of their expected lifespan. And so we thought, well, we really need to start looking for something else. A sensible person would probably just go to the next iteration, say something like the BD Vax Lyric, 3 color, 12 laser. But we're a research hospital, and we decided that we wanted to really try and push the limits. So the format that we've decided on is the ID7000 Sony Spectral. The models that we're using in the lab are the 6 laser, which are really theoretically capable of doing 50 or more colors. So a lot of people said, have you lost your mind? And I thought of JFK's really inspirational quote from Rice University. We do these things and others, not because they are easy, but because we thought they'd be easy. Pretty sure he didn't say the last bit. So now let's just look again at the ID7000. We had a lot of experience running Sony. We'd had the Sony SP6800 and the Sony sorters. So that's how we sort of ended up on the Sony route, in case anyone's wondering why not Scitech. It was just that we happened to be Sony-centric early on. Anyway, so if we take the lid off that, we can see that these spectral cytometers are really just like any other flow cytometer on the front end. You've got the sample injection port. You can interrogate a stream of single cells with a variety of different lasers. But really the business end of the spectral and the difference between conventional flow and spectral is the way that the data is handled downstream from your flow cell. So for example, if we look at this 320 nanometer laser deck, rather than a series of filters and mirrors and looking for, maximas of, fluorochrome excitation, we actually look at the entire spectrum of the output from that laser line. So we have 35 PMTs measuring the emission output from these cells between 375 nanometers out to about 840 nanometers at about roughly 10 to 11 nanometer intervals. So really the power of spectral comes from the fact that we have multiple lasers and all of these different detectors, so that for any sample, we are measuring the sample, we're getting data from every laser and every different PMT. So for each single color control or cell, we end up with 182 fluorescent data points from these 182 different detectors. So if we look at something like our old friend PE and run that on this, we end up with this sort of line of cells. So if we, in the sort of math speak, I guess, we would call this our measured vector. I like to think of it as this is the shape of PE, and it's invariant, right? So it's not going to change from day to day. And so this is the overall spectral shape of PE. Now, this is quite different to what we would do, or the data that we would get from our Canto 2. So on our Canto 2, we would throw out nearly all of the fluorescence information and really just focus on the blue laser, and using filters and mirrors, be able to count photons in a very discrete region around the emission maxima for PE. And this would be a 42 nanometer window and would have a single PMT, which would count photons in that region. So comparing for PE on the spectral, we had 182 data points. On the Canto 2, just one. Now, because of this, we can't use simple subtractive compensation. And instead, we need to use a linear algebra term, which at first looks a little bit frightening, but really isn't. And this was invented almost 200 years ago by Gauss. And it really, what it does is that we take our spectral data, our shapes of each of our individual cells, and we compare that to the individual shapes of all of the single color controls. And we ask, the equation asks, what combination of our single color control shapes gives us the best match for the shape that we've observed in our cell. And that gives us omega hat. Oh, I'm so sorry. That gives us omega hat, which is our output, which is just parametric data that we can plot as dot plots or histograms, or however we're used to plotting flow data. So for our spectral references, and I sort of already alluded to it, the real power of spectral is that multiple fluorochromes are excited by different lasers. So we get the combined information from different laser lines for each of the different fluorochromes. Now, this added information is really good, but one thing that we cannot escape either in conventional flow or in spectral flow is the problem of spectral spillover spreading. Now, I'm not going to go into any details about how it's measured or what the consequences of it are. But really, for us in our panel design, we decided that really what we wanted to do, if we had, say, 45 or 46 potential colors that we could use, where does all of the potential spillover spread occur? And can we eliminate those fluorochromes from our matrix? And that's exactly what we did. So this is a spillover spread matrix. All you need to know is that if you've got red, you've got spread. So I'm a fairly simple sort of guy. I just thought, let's just get rid of all of the ones that introduce a lot of red and see what we have left. And so after doing that, we ended up with 38 colors that we felt gave us a reasonable high number of parameters, but also reduced a lot of the red out of that matrix. So then we decided we wanted to fit as much of our immunophenotyping panel, the standard panel that we run every day on new or relapse diagnostic samples, and try and fit as many of those into one single 38 color tube for the spectral. Because we didn't like, we wanted to make it even more difficult for ourselves, we decided that we would only use commercially available antigens and conjugates. And we would not make any custom conjugates just because of the issues that might come on later on in trying validating tests and things with the availability and stability of different custom conjugates. So we just wanted off the shelf, where possible, ASR reagents that we could use in our panels. So we ended up with this 38 color panel that we started to work with. Now, and amongst this as well, we had some other constraints. For example, like in FTSE, we knew that we had to have TDT on there because we use this Supertex mixture of TDT clones, and that's only available in FTSE. So there were a few parts in our panel where we were constrained by, we want a particular clone or particular color in this place. So that sort of limited our ability as well a little bit. But then we just sort of went through the iterations of trying to put together a 38 color panel that we could use for immunophenotyping. We ended up with 31 surface markers, 5 intracellular markers, and live, dead, and nucleated markers. And we'll get to these in just a minute. But really what this did was to give us a palette of colors that we could use for the development of many other panels coming on for our Bs, Ts, and AMLs. And we'll show you some of those towards the end of the talk. So we had this, and the next thing to do, obviously, is to start adding antibodies to tubes and actually measuring the spectral outputs. So this sort of slide belies really all of the work that went into this. So, for example, our most difficult to place antigen was CD33. And you would think, well, that should be relatively easy. But we went through between 14 and 16 different fluorochrome combinations to get that in the right place. And we did that because we knew in our AML MRD panel, we needed certain characteristics of CD33 expression to be able to be distinguished. And we didn't want to have necessarily CD33 different in different panels. We want to try and keep the backbone as big as possible between each and not have multiple different fluorochromes for different antigens. So we started running these and did the unmixing. We have our 38 color panels, do the unmixing to give our Omega hat and give us our parametric data and start plotting data. So here's just forward and side scatter. I'm not going to go into a great deal of detail about the panel design, but I will talk for a few minutes about our efforts to try and identify what we call our correction factor for our Canto2 data. And we decided to put our sort of common markers, as it were. We use DRAC5 as our nucleated marker. We'll just zoom in on this. We use DRAC5 for our nucleated marker, zombie NIR as our live dead marker, and in between that, we wedged CD45. So nearly everything in the panel that we're interested in are going to be nucleated, they're going to be alive, and nearly all of them are going to express CD45. So they're going to be very common antigens on there. And so we thought we would save, essentially, have these out on the red laser. There's very little sort of flow back into these higher energy, low wavelength lasers. So we kept these lasers for all of our specific antigens and things, if that makes sense, and tried to keep our sort of kind of housekeeping fluorochromes down on the red laser. And the reason we did this is because on our Canto2, we have yet another tube that we run for every time we run an MRD. So our MRD tube, which is that backbone plus the add-on markers for the immunophenotype identification, that gives us the numerator for our MRD calculation, but then we need to be able to work out what is the denominator. And we use our Cyto13 tube, which is for a correction factor, to correct our total events recorded into a mononuclear nucleated cell count, which is our denominator for our MRDs. So the way that would work on our Canto2, there's a little picture of a Canto2 here. We would record the same number of events, 10 or 20,000 events for both the tubes, and a tube stained with Cyto13, which is just a nuclear marker with 45 and glycophorin A. And so we just compare the two populations on the two, and that those ratios should be the same. And so we can generate a correction factor just by rearranging that equation. So we do that for every sample that we are doing our MRD analysis on. So we wanted to build that into every panel that we had, so that we'd automatically have our correction factor built into our panels that we're designing for both immunophenotyping and MRD analysis. And so the way that the gating on that would work, so we've got our scatter plots, we'll get rid of doublets, and then we plot the zombie versus DRAC, so dead versus nucleated, and we have this live nucleated cell gate, which now becomes our denominator for any of our MRD calculations. We don't need to do any correction factors or things, that's already built in. So then we can compare, obviously, going back to the spectral ribbon plots. By doing this, we dramatically improve the output of our data. You can see a lot more complexity in this live nucleated cell gate compared with our dead cells or just the total cells all over. So then we take our live nucleated cell gate, and then we can start plotting this just as side scatter versus 45. We can see our nice lymphocytes, monocytes, and granulocytes. Start adding in various like CD19 for B cells, both surface and intracellular, so 79A for B cells, different T cell markers, and different myeloid markers, so monocytes, granulocytes, intermediate monocytes, NK cells, etc. And now the other thing that really is quite remarkable with spectral is the ability to identify these autofluorescent populations and actually deal with them. So anyone's done any flow or used to these sort of rocket sort of plots or whatever we'd like to call them. And it's really due to autofluorescence in the sample. So we took a really old cruddy sample and ran it, which would have a lot of dead cells and autofluorescence. So we're able to gate on an unstained population and identify the fluorescence of this autofluorescence. And we can define that autofluorescence as its own measured vector or shape, and then add that to the unmixing algorithm. And on the ID 7000, we can identify 7 different autofluorescent populations. But in this case, we just have one. So then we can go back and do the unmixing, either with the 38 colors of our panel or 38 plus the autofluorescence channel, 39 colors. And you can see we get a much better resolution of our plots. Now, not only just on the wavelengths where you would expect that autofluorescence to be, but across the board, by doing the autofluorescence finding, we're able to tighten up all these populations. And it kind of makes sense because there's extra fluorescence in there. And so the algorithm's trying to say, where do I fit that extra fluorescence? And it's got to go somewhere. So if it knows where to put it, that is actually just due to autofluorescence. It does improve the overall quality. So for all of our assays, we now do an autofluorescence finding as part of our initial stages of our data cleanup. So one of the other things that we decided to do was also we needed a software package that could handle this high dimensionality data. At the moment, we're using both FCS Express 7 and also Infinisight. Infinisight is particularly useful for us because one, it handles these really high complexity data very easily. It also speaks to our medical record system, EPIC, very nicely as well, so there's no issues there. But also I'm pointing at the wrong thing. We could also use the principal component analysis as automatic population separator. So this is just let's go back. This is just a normal peripheral blood. You can see CD4 and CD8 T cells, B cells, monocytes, granulocytes, and NK cells, all separated just based on its own separation. So it makes gating and analysis of these much easier. This is just an example of a B leukemia at day 15 bone marrow, and you can see the blasts are in red and they segregate near these normal B cells, and it's very easy to identify and gate these leukemic blasts. Now, we really have to sort of address the elephant in the room. I'm from New Zealand, so we actually say taniwha in the hot tub, but it's the same thing. And it's really about data analysis. So if you think about it, and I know the math isn't right here, but it's just for effect. So on the ID 7000, we've got 182 fluorescent detectors. We record height, width, and area, and time and a bunch of other things. So if you compare it to the Canto 2, our data file for the same number of events is going to be at least 50 times bigger, right? So how do we start to manage some of these data files? So one thing that any cytometrist would do would be first to look at thresholds. So one thing that we would set our forward scatter threshold to make sure that we're not recording just all of the debris out of the sample, right? So you could draw a threshold like this and say, okay, anything below that line on the forward scatter is just debris and we don't care about it. Now, we didn't want to go too nuts with this because a lot of leukemic cells can be very small. So we didn't want to sort of just keep creeping up that forward scatter threshold because there could be a danger that some of the very small blasts might get chopped off by doing that. So then we decided to look at some of our early gating strategy. So if this is, for example, a sample that was relatively old, so it'd been 3 or 4 days just sitting around in the lab, and we run it. So it has a lot of debris. And here are our live nucleated cells, lots of debris because the sample has been sitting around. And if we take that sample and just randomly gated 3 populations, so we had mature B cells, helper T cells, and monocytes, and we sort of see this ratio down here. Now, I do apologize. Beijing keeps changing this live nucleated cell gate to the Mead gate. I keep changing it back, but sometimes it slips through. So anyway, so live nucleated cell gate over here. But if we look at the spectral output from this, we can see here is just a scatter plot, and we can see essentially 2 populations here, right? So if we now move over and then look at the live nucleated cell gate, we see that the live nucleated cells, these are DRAC5 positive, zombie negative, they are sort of this top line, whereas what we're calling the debris, which is essentially the DRAC5 negative population, is down here. So what is it what are we looking at really with the DRAC5? Sorry, I'm still confused. And if we look at the emission maxima for DRAC5 off that red laser, we see that it is located on the PMT22 or channel 21, and it's measuring between around 690 and 700 nanometers. It's the maxima for the DRAC5 signal. So we can, on the instrument settings, we can actually set a second threshold based on the single PMT out of the 182 PMTs to be able to try and remove some of that DRAC5 signal from our sample, as it were. So this is an example of what it would look like. So here's where we've got the sample just using the forward scatter threshold. And then if we set that single laser PMT21 threshold to 10%, we maintain all of our not live nucleated cells, but we get rid of all that debris out of there. And so Beijing went through and did all, due diligence and looked at various threshold settings. And we decided to set our market on 10% for our PMT21 red laser, the DRAC5 region. So then if we now compare that, so we now eliminate all of this debris, and rather than 10,000, so we recorded the same number of events on that sample, now we've got 3 times as many of the live nucleated cells that have been recorded. And then we can just do that same simple gating, and we didn't do it very well, but we can compare either the threshold set to 0 or 10%, and the actual ratios of these cells remain exactly the same. So we're not losing any of the cells, we're actually just recording events that we're actually more interested in rather than all of the debris. So our practice at the moment is that we'll record 2 samples for each one. We'll record about 50,000 ungated events, meaning that we don't set the threshold. Then for our mini events, for our MRD calculations, we will actually set that threshold on PMT21 and red laser to 10%, and then record most of it. This is just a sort of a safety measure to make sure that we can compare and we're not accidentally losing anything out of any specific sample. So at this stage, we've got our 38 color immunophenotyping panel pretty much nailed down. It's not perfect, but Winston Churchill said, perfection is the enemy of progress. And so we decided this is going to work, and it works really nicely. And we knew that because Beijing has been running lots of samples, and this is a little outdated at the moment, but we've run over 37 Bs, 22 TALLs, 23 AMLs, and a whole bunch of MPBs. And compared that with the Canto 2 data that's coming out of the clinical IP lab, and we've got 100% concordance between all of those different samples. So we decided that it's time now to buy 2 more ID7000s. We cleared out a bay in the clinical lab and installed, I don't get out much, we called them Isaac and Newton, and there they are in the clinical IP lab. And we've, because I'm also technical director in that lab, I'm working really closely with Laura Key, who's taken on this monumental project. Don't think she really knew what she was getting into. But anyway, she's really taken to these like a duck to water, and it's great. And so just a very small part of our assay validation plan is to run 20 Bs, 20 Ts, 20 AMLs, and compare that, run them on the same day on the Canto 2 and on both of the ID7000s, compare the data. In fact, our validation plan, or even the checklist for our validation plan, is more than 3 pages long. So that's just telling us what needs to be in the plan. So it's going to be probably well over 1,200 page document by that time we're done with this validation. So at the moment, the panel design, which is done in my lab over on the research side, is done for the immunophenotyping. The clinical validation is well in progress now. Laura's done all of those different samples that we need, but we now need to go back and do cocktail stability, interoperator, variability, all of that stuff that we will do. We're in the process of developing a TAL MRD, the Bs, and the AMLs and lymphoma panels yet to be started. So now if we go back and look at our current workflow in our clinical IP lab, doing it on the Canto 2, so we see have this quite extensive workflow, even just on day 1. So in the very near future, this is going to be replaced by our spectral workflow, where we now have a sample. We run a single tube for our immunophenotyping. Then we would do lineage assignment, and we would run, for example, if it's a BALL, we would just run our BALL MRD panel. And then for every follow-up, any BALL patient would get the same tube, but there would no longer be the need to be able to identify a patient-specific follow-up tube, which will make life a lot easier for our flow group. So in the last few minutes, I'll just show you some of the where we're at with our MRD panels. So again, as I mentioned earlier, when we went down to 38 colors by trying to get rid of a lot of spectral spillover spread and stuff, I think we're a little bit, too worried about that. In fact, we can go up to 43 colors quite easily. So our current T panel is the 39 colors. And I'll just give you an example of a patient that came through recently. So the immunophenotyping indicated that was a TALL. So then we would run the TMRD panel. In our TMRD panel on the Canto 2, we actually use 6 of the channels to identify the blasts. And this is essentially our backbone. So it's got cytoplasmic and surface CD3, CD45, CD5, 1933, and DR, or all negative markers. Then we have these add-on markers, which we use again for identifying our leukemia-associated immunophenotype. So this is just, I've put a little Canto 2 down in the corner so you know what you're looking at. So this would be, for an example, here are the blasts here. They've got variable cytoplasmic 3, the very dim for surface 3. They've got dim 5 and dim 45, and completely negative for 1933 and DR, so that they look like blasts. And then we add those add-on markers, and you can see that these guys express CD34 and a 7. There's a subpopulation that's 1A positive. That's got this bizarre CD4 and CD8 double population, but again, just a partial. They're 99 positive, but very limited TDT expression. So again, we used our sort of same color palette that we're using, and all of the yellow markers indicate the difference between our immunophenotyping panel and the TMRD panel. So we can add it in a whole bunch of different, for example, NK markers and a few different T markers as well. This is our acquisition template for the TMRDs. Again, there's a little ID 7000 down in the corner. So if we compare, on the top line is the Canto 2 data, On the bottom line is the ID 7000, and you can see they look pretty much the same. We're getting probably a little bit resolution, but a little bit better resolution on the ID 7000 as you'd expect, but with one caveat. Usually we cannot get these samples until a day or two later to be able to do in the research lab, because we need to make sure that they aren't needed for any clinical testing before they're released for these sort of development work. So keep that in mind, but everything looks pretty similar. And then if we move on to those add-on markers, we can see, again, they look very similar. We're getting a little bit nicer definition of the TDT. Again, we have this 1A subpopulation and this weird 4 and 8 double population and a part of the population. Just finally, we'll go on. We've now got a 40 color BALL MRD panel. And again, we're using the same template. The yellow ones indicate the differences that we've added to our panel for the Bs. I will show you 2 patients very quickly because they're quite interesting. Patient 2 here had overexpression of 73. And so again, if we compare the data that we saw on the Canto 2, again, we use the backbone to identify blasts. And we can see here, they've got CD19 DIM, 45. They overexpress CD10, but there's a dual population. Some of the blasts express 34 and some of them are negative for CD34. And then if we run those, again, our add-on markers looking for the leukemia-associated immunophenotype, the best panel to run for this guy would be 38, 73, and 20. They've got DIM 38, overexpression of 73, and they have high levels of CD20. So this patient would be relatively easy to follow. And it looks the same both in the CD34 negative and CD34 negative populations. So then again, we'd run that same sample on our ID 7000. Here's the acquisition template. And again, comparing on the top, the Canto 2 to the ID 7000 below, and we see exactly the same sort of patterns. One caveat here is that we ran a bone marrow on the Canto 2. There wasn't any bone marrow left, so we grabbed a PB from the same day. But we can see the same distribution of blasts. If we look at the leukemia-associated immunophenotypes, it looks exactly the same between the Canto 2 and the ID 7000. And of course, the real advantage is we've got a lot more markers on the ID 7000, including CD9, 24, 86, DR, and 81 are all aberrant. And also we're able to identify that this other population that popped up are actually NK cells and nothing to do with the leukemia. So then finally, we have our patient 3. This is a 38 DIM population. So it looks fairly similar on our backbone markers, with the exception now that these 45 DIM population, they're uniformly 34, but have variable CD10. And looking at the other markers, we ended up using exactly the same panel because it's 38 DIM, it's got a variable expression of 73 and variable 10, but they're dead negative for CD20. So again, they'd be easy to follow using the same panel, but they have quite a different immunophenotype that we'd be following. So again, moving to our spectral, here's the data acquisition on the ID 7000. And again, the comparison between the Canto 2 and the ID 7000. So again, looking at this 34 positive population, variable CD10, just like we saw on the other one. Again, we see variable expression of 73 and 10, almost negative CD38, and very dim 20. So just like on the Canto 2. But again, on the ID 7000, we've got expression of CD9, we've got overexpression of 58. I can't even see what that says. Anyway, there's other markers, but also quite interestingly, we have ectopic expression of 66C on this, and also expression on our blasts of both 13 and 33 that we didn't pick up on our Canto 2. So this one would be a relatively easy one to follow. It also has overexpression of CD123, which would be useful for us because if they fail induction, then they'd be eligible for one of our CD123 CAR-T protocols. So again, in our near future, we'll end up with a much more streamlined, much more data-intense workflow for our MRD follow-ups. And then finally, I'd just like to really thank the people involved in this. Primarily Beijing Liu has done a lot of the work on the assay development or the panel development. Other members of my group include Mo and Zhao Hong. Our hematopathologists have been instrumental in choosing all of these antibodies and things that are in it, particularly Jeff Flo, our division director, Priya Kumar, our newly minted medical director, Martha, Gabriella, and Yen. I really want to thank Jeff Clapper from Sony, who was really helpful in our sort of early efforts in getting this all running. And of course, our clinical IP lab, who's led by Christy Embry, our manager, and supervisor Celine, Laura, Lisa, Chris, Sabina, Pam, Rat, and Pam. Thank you. Oh. Sorry if I went a bit long.
No, you're great. Thank you so much, Paul. That was wonderful. We do have some questions. Yeah.
Thank you. Hey, a great talk, but I was curious, have you had to deal with a lot of unmixing errors in a panel this size, and how would you account for those, especially as you translate to a clinical lab?
Yeah, that's a really good question. Yeah, the unmixing, a couple of things I'll say about that. The biggest influencer on the unmixing errors in there are usually from that red laser with our DRAC for our nucleated marker and the zombie. If we get that sorted out early, then everything else generally falls in line. So if we do see an unmixing error, it's almost certainly due to one of those two, DRAC or the zombie. The thing that has helped that the most is being really, really carefully titrating not only those dyes, but all of the antibodies. We've found that now that we've got it locked into, titrating carefully, not only titrating the antibodies on their own, but how they interact with each other, make sure that we balance those well, then the unmixing seems to become, there's very, very little to do with the unmixing. One caveat I would add to that, and I just want to be completely honest here, that say, for example, if a kid comes in and they've got a BALL and they've got screaming hot CD10, which is not uncommon, that can cause problems, right? It's not going to change our clinical diagnosis because that, it's not going to unmix it that badly that you're going to go, was that really a T or an AML? No, there's no question, right? So, but we would want to try and either tighter down the 10, which is on BB480, or do something for that particular sample when it came back through. But but generally, if they're really screaming hot with treatment, they tend to go down anyway. They don't always stay that, and they're not that prevalent. Like, so if the kid comes in and they've got like 80-90% blasts and they're all got, super bright 10, then yeah, that can really affect our unmixing. But in general, if we're titrated well, it's good. Sorry. Thank you. I can talk more about it later.
You sure?
Great talk. I have a question. Do you need to run here? I'm sorry, too loud.
Oh, sorry. So,
so my question is, do you need to run single color control on a daily basis? And if you run the single color control on a daily basis, you use beads or use the cells for single color controls?
We do not run single color controls on a daily basis. That would be rate limiting, I think. At the moment, we've done this a lot over the last couple of years, and both in the translational lab, but also over the last sort of 8 months or so in a clinical lab. Yeah, we've sort of, we're trying to still work out just exactly how long we need to do. So for nearly all of the fluorochromes, we've decided that every time we get a new lot number and we run a new single color control, and then add that to the library, and that becomes into the unmixing. Now that becomes really, really important for things like the dyes, like DRAC5 and other things, and even also with some of the antibodies, right? Not a lot of companies have a 320 laser, so if they're QCing their product, no matter who it is, I'm not going to say anyone in particular, but we have noticed that in some batches, we see some signal in the 320 nanometer range that maybe they didn't even look at because they don't have a 320 nanometer laser to do their QC. And so that is very important to make sure that that is included in your unmixing if you're using that lot of antibody. I didn't really answer your question, but no, we do not run our single color controls every day. We do it each lot number. The only problem with that is that BD, if they do their opti builds, every tube is a different lot number. So we're trying to work out that. But yeah, definitely not every day. And we have not seen a problem yet. So.
Paul, as always, fantastic presentation. I have two questions for you. One's around stability and then one expands on controls. So what is the tube matrix that you're using? Because I know you had alluded to it that you're not running the samples fresh, right? And so obviously stability is a concern. So what's the tube matrix that you're using or preferred? When are you putting it on the instrument in terms of how long post-collection is it sitting around? And then controls. So you spoke a little bit about the single color controls, but what about like FMOs or batch controls? And did you have any surprises when you transitioned from a traditional cytometer onto a spectral when you implemented those controls? Thank you.
Yeah, sure. Hopefully I can answer all of those. Yeah. What was the first bit? Stability. Yeah, so I see you're alluding to like fluorochromes, like tandems that might fall apart and things. Stability of the sample. Oh, sample stability. Sorry. Okay, so yeah, so I might have misspoke there a little bit. So we're sort of like the translational IP and a clinical IP are two different entities, and so for us to get samples to run and essentially on the research side, we have to know that those samples are not going to be needed by either molecular or cytogenetics or anyone else. So there is usually a delay in us getting it. So we just end up getting, usually it's not bad. Sometimes we can get them on the same day, but sometimes we have to wait at least a day or two before, say, a bone marrow might get released. In the clinical lab, that's not going to happen, right? So we would just get the marrow or whatever comes in. We would just draw cells to run on the Canto at the same time, run on the because they have to be run at the same time anyway, part of our validation. So it's really just over on the other lab where that becomes an issue. In general, what we do is that any leftover cells immediately go to our biorepository where they're fivefold and banked. And so we don't generally use a lot of stabilization solutions. We do have a lot of outreach with like our global partners and stuff. So we're really interested in looking into that. So if anyone's got any sort of magic bullet for that, I'd be really interested. But within the hospital, nearly any cells left over immediately go to the biorepository, are fivefold and cryopreserved.
Yes, very impressive work. I'm highly impressed about your identifying 7 different populations that have different autofluorescence pattern. So I have 2 questions. So first question is whether this is identified from normal sample or patient sample. And the second one is, have you ever find some patient sample have a different pattern? Maybe sometime it has a strange autofluorescent population.
Oh, yeah, no, great questions. So again, I probably misspoke. So on the IDU 7000, you've got the option of gating 7 different autofluorescent populations. Often you don't need to do that. We can just do like 1 or 2 if they're really, strikingly different. The corollary to that is sometimes you don't see an autofluorescent population at all. And so what we've found is it actually helps, even if you don't see like a really obvious glaring autofluorescent population, just gate on your bulk population and the unstained. And just say, that's my autofluorescence of my, it's going to be dim, but adding that to the unmixing helps a lot as well. So just the endogenous, not crazy autofluorescence, but just the endogenous autofluorescence of the bulk population can be used as well to help for the unmixing. And the second part was? Yeah. So sometimes, Not so much in spectral, but we are aware of it. Like so some of our AMLs, they can end up with these either, you know, excessive basophil populations or eosinophils or dendritic cells and things. And particularly basophils and eosinophils will have a very characteristic autofluorescent signature. So yeah, we do see those, and we're able to gate based on autofluorescence of those populations, not only to get rid of it, but to actually use that channel as another color. Looking at this very specific autofluorescent signal of those. In general, though, what we've been doing is actually just taking those samples, and because they will often have some sort of, you know, like we do whole genome, whole exome, RNA-seq on every patient that comes through, every sample. So, you know, when we get differences with a molecular and flow MRD, then we'll, you know, maybe go back and sort the cells and say, you know, maybe the signal's coming from that basophil population or something, which is something that's happened recently. But yeah, we should be able to definitely see those on as autofluorescent